Good day, students. You are welcome to another video lecture on catabolism of amino acids. So today we are going to be discussing about amino acid catabolism. So any amino acids in excess of biosynthetic needs of the cell are rapidly degraded. So as we know, excuse me. As we know, amino acids provide nutrients to the body. They are building blocks that are used for bodybuilding. So they also provide energy. Uh, the energy they provide is almost equivalent to that provided by lipids. So those amino acids that are constituent of the proteins after digestion, if the cell does not need those amino acids for energy, okay, they are going to be used for biosynthesis, okay? They are going to be used to build the body. But if the body needs it for energy, they are going to be degraded. So how are they going to be degraded? There are two ways in which the amino acids are degraded. So the first phase of catabolism involves removal of amino groups. Usually, if you look at the general representation of amino acid, there is amino functional group. Okay, there is amino group, and the remaining parts of the amino acid is called the alpha keto acid or carbon skeleton. So usually the amino groups are removed by a process that is called transamination, and also there is also oxidative deamination that you can also use to remove the amino acid and the amino group. So after that, it means the amino group is now in the form of ammonia, and the corresponding remaining parts of the amino acid is called alpha keto acid, or is called carbon skeleton. So a portion of the free ammonia is excreted in the urine, but most of it is used in the synthesis of urea, which is quantitatively the most important route for disposing the nitrogen from the body. So the role of body proteins in this transformation involves two important concepts: the amino acid pool and the protein turnover. So how is this amino acid pool formed? The amino acid pool is supplied by three sources. Number one, the amino acid provided by the degradation of endogenous that's body proteins, most of which are reutilized. Number two is amino acid derived from exogenous, that is to say dietary proteins. And the third one is non-essential amino acid that are synthesized from simple intermediates of metabolism. So how is now this amino acid pool depleted? So it's number one, synthesis of body protein. Number two, consumption of amino acid as precursors of essential nitrogen containing small molecules. And number three is conversion of amino acids to glucose, glycogen, fatty acid, and ketone bodies, or oxidation to carbon dioxide and water. So there is also what we call protein turnover. When we say protein turnover, it means we are now talking about the amount of protein that is now produced, okay, by the body, and it's also constantly undergoing degradation. So most proteins in the body are constantly being synthesized and then degraded. They also undergo degradation, permitting the removal of abnormal or unneeded proteins. So the rate of protein synthesis is just sufficient to replace the protein that is degraded. This process is called protein turnover and leads to the hydrolysis and resynthesis of about 300 to 400 grams of body protein each day. So there is what we call proteins that live shortly, short-lived protein. For example, many regulatory proteins and misfolded proteins, which are rapidly degraded. They have their half-lives measured in minutes or sometimes hours. There are proteins that lives that have half-lives of days to weeks, which are called long-lived protein, and they constitute the majority of the proteins in the cell. And we have also structural proteins such as collagen, which are metabolically stable and have half-lives measured in months or even years. So there are now these steps that are involved in in catabolism of amino acid. We look at transamination reaction, we look at the oxidative deamination, we also look at transport of ammonia to liver and also urea cycle. So how is nitrogen is removed from the amino acids? Now we're going to look at how the body now remove nitrogen from the amino acid. So the presence of amino group keeps the amino acid intact. It means free from oxidative breakdown because the amino group is on it. So now if the body now remove the amino group, it means now the carbon skeleton is going to undergo oxidation. So removing the amino group is essential for producing energy from any amino acid and is an obligatory step in the catabolism of all amino acids. So once the amino acid is removed, this nitrogen can be incorporated into other compounds or excreted as urea, with the carbon skeletons being metabolized to produce energy or other needful products in the body. So these two processes involve nitrogen removing from amino acids, transamination and oxidative deamination reactions, but that ultimately provide ammonia and aspartate, which are the two sources of urea nitrogen. So if you look at now this amino acid, there's amino acid and their corresponding alpha keto acids or carbon skeleton. So if you look at alanine, the corresponding when amino group is removed from alanine, the alpha keto acid or the carbon skeleton that will be remaining is called pyruvate. Okay, for aspartate, when the amino group is removed, you have oxaloacetate. So amino acid here, and we have on the left and on the right we have the alpha keto acid or carbon skeleton. So for glutamate, you have alpha ketoglutarate. So all other amino acids are either converted into the three above of alpha amino acid or alpha keto acid. Okay, so this is what is happening here. If you look at this diagram, we have keto acid here. Okay, you have keto acid here, and we have amino acid. So we have the amino acid now will be a substrate that donates the amino group, and the keto acid now here, which is now the amino group acceptor. So when that happens, you have the corresponding keto acid being produced, and also the product amino acids now produced. So transamination reaction now. Funneling of amino acids to glutamate, we have the first step now in the catabolism of the amino acid is the transfer of alpha amino group to alpha ketoglutarate, which is now producing an alpha keto acid and glutamate. The alpha ketoglutarate plays a pivotal role in the amino acid metabolism by accepting the amino groups from most amino acids, thereby becoming glutamate. Okay, so alpha ketoglutarate receive amino groups from most of the amino acid, and it becomes what? Glutamate. So alpha ketoglutarate forms glutamate when it accepts amino groups from amino acid. And this transfer of amino groups from one carbon skeleton to another one is catalyzed by a family of enzymes that are called aminotransferases or transaminases. And these enzymes are found in the cytosol and mitochondria of cells throughout the body. So all amino acids, with the exception of lysine and threonine, participate in transamination reaction at some point in their catabolism. So this is generally what is happening. We have two aminotransferases. We have alanine aminotransferase and aspartate aminotransferase. So if you look at here, alanine is the amino donor, and alpha ketoglutarate is the amino group acceptor. So when alanine provide the amino group, the alpha ketoglutarate will now form our glutamate and the corresponding alpha keto acid, which is pyruvate from alanine. And the enzyme that catalyzes this reaction is alanine aminotransferase, or ALT, and it requires pyridoxal phosphate, PLP, as cofactor. So we have aspartate aminotransferase. Now for aspartate aminotransferase, we have the aspartate now provide the amino group, while alpha ketoglutarate now accepts the amino group to form glutamate, and aspartate now is converted to the corresponding alpha keto acid, which is oxaloacetate. And the enzyme here is aspartate aminotransferase, or AST, and the requirement is what? PLP as the cofactor, pyridoxal phosphate. So two enzymes are involved in transamination, alanine aminotransferase and aspartate aminotransferase. Now, elevated levels of plasma levels of aminotransferase indicate damage to the cells rich in these enzymes. For example, if physical trauma or disease process or disease condition can cause cell lysis, resulting in the release of intracellular enzymes into the blood. So when there is high level of this, elevated levels of these enzymes, it means there is tissue damage or cell lysis, as in the case of, let's say, liver. When you now have liver diseases, if it means there is maybe active degradation of liver cells and you have these enzymes being elevated in the plasma. So two aminotransferases, AST and ALT, are of particular diagnostic value when they are found in the plasma. They indicate, high level of them indicates disease condition. So oxidative deamination is glutamate produced by transamination can be oxidatively deaminated. In contrast to the transamination reaction that transfer amino groups, oxidative deamination reactions result in the liberation of the amino group as free ammonia. These reactions occur primarily in the liver and kidney. So we have this enzyme called glutamate dehydrogenase, which is now the enzyme that catalyzes the reaction when you have the amino groups of most amino acid are ultimately funneled to glutamate by means of what? Transamination with alpha ketoglutarate. So now glutamate is unique in that it is the only amino acid that undergoes rapid oxidative deamination, a reaction catalyzed by this enzyme, glutamate dehydrogenase. So therefore, the okay. So now, this glutamate is unique in that it is the only amino acid that undergoes rapid oxidative deamination, a reaction that is catalyzed by glutamate dehydrogenase. Therefore, the sequential action of transamination and the oxidative deamination of that glutamate, regenerating alpha ketoglutarate, provide a pathway whereby the amino groups of the most amino acid can be released as ammonia. So if you look at this diagram, it's now trying to explain the removal of amino group from an amino acid. So here, if you look at here, we have transamination, which is catalyzed by aminotransferases, and you have amino group of amino acids are being collected or are transferred to carbon skeleton. For example, is here alpha ketoglutarate to form the corresponding alpha keto acid and glutamate. Now, in oxidative deamination, it is now glutamate dehydrogenase that now collects the amino group from the glutamate and to form alpha ketoglutarate and this reaction is now an ammonia, and this reaction is now catalyzed by glutamate dehydrogenase, which requires NAD plus or NADP plus to form NADH or NADPH. So this ammonia that is produced, it goes to urea cycle, and you have this amino acid that produced, and you have your carbon skeleton here from the amino acid that is the corresponding amino acid that donates its amino group. So transport of ammonia to liver. Two mechanisms are available in humans for the transport of ammonia from the peripheral tissues to the liver for its ultimate conversion to urea. The first uses glutamine synthetase to combine ammonia with glutamates to form glutamine, and which is a non-toxic form of the ammonia. So glutamine synthetase combine ammonia with glutamate to form glutamine. It is now glutamine that is transported via the bloodstream to the liver, where it is being now cleaved by glutaminase to produce glutamate and free ammonia. And the ammonia is now converted to urea. So the second transport mechanism involves the formation of alanine by the transamination of the pyruvate produced from both aerobic glycolysis and metabolism of the succinyl coenzyme A, generated by the catabolism of branched-chain amino acids such as isoleucine and valine. Alanine is transported by the blood to the liver, where it is converted to pyruvate again by transamination. So the pyruvate is used to synthesize glucose, which can enter the blood and can be used by muscle, which is through a pathway called glucose-alanine cycle. So for urea cycle, urea is the major disposal form of amino groups that are derived from amino acids and account for about 90% of the nitrogen-containing components of urea. One nitrogen of the urea molecule is supplied by free ammonia and the other nitrogen by aspartate. You should not forget that glutamate is the intermediate precursor of both ammonia through oxidative deamination and by glutamate dehydrogenase, and aspartate nitrogen through transamination of oxaloacetate by aspartate aminotransferase. The carbon and oxygen of urea are derived from carbon dioxide as bicarbonate, and urea is produced by the liver and then is transported in the blood to kidney for excretion in the urine. So these are the reactions that are involved in urea cycle. Number one is the formation of carbamyl phosphate, and this is catalyzed by carbamyl phosphate synthetase 1. And this reaction is driven by cleavage of two molecules of ATP. And ammonia is incorporated into carbamyl phosphate, is primarily provided by oxidative deamination of glutamate by mitochondrial glutamate dehydrogenase and producing nitrogen atom that is derived from this ammonia becomes one of the nitrogen of urea. So carbamyl phosphate synthetase 1 requires N-acetyl glutamate as positive allosteric activator. There's another enzyme called carbamyl phosphate synthetase 2, which is in the biosynthesis of pyrimidines. Number two is formation of citrulline, and this is catalyzed by ornithine transcarbamylase, which is now transfer of carbamyl phosphate to ornithine. Okay. So the third reaction, okay. Okay. Now for the third reaction, Before now, the reaction that produce citrulline, the reaction product which is citrulline from the previous reaction, okay? You have ornithine being that received so ornithine that received the carbonyl phosphate, okay, it forms citrulline, and now this citrulline is transported to the cytosol. Note that ornithine and citrulline are basic amino acids that participate in the urea cycle, moving across the inner mitochondrial membrane via a co-transporter. So ornithine is regenerated with each turn of the urea cycle, much in the same way that oxaloacetate is regenerated by the reaction of the citric acid cycle. So the third reaction, number 3, is a synthesis of adenosine. This adenosine synthetase is the enzyme that combines citrulline with aspartate to form adenosine.